as you know, drought and heat, these are the major abiotic stresses in case of it. And then for saline areas, salinity areas, areas which are having salinity problems, so you should have salinity tolerance. But these are the broad areas. Then there are other areas also. Is a hybrid wheat breeding. We have been working on hybrid wheat breeding. So there also we are using these tools of marker state breeding. And then there can be other traits also. We will discuss quality traits also. But I will not be discussing all these things. I will be discussing some of the basic things. Okay, basic methods, basic methodology remains same. So we will discussing some of the things. And for that, whatever marker state breeding or genomic state breeding we go for, we require genomic resources. And in wheat, so far we were depending mostly on SSR markers, microsatellite markers, and quite a good number of microsatellite markers are available with us. We can work with these microsatellite markers, and then now we can go for SNP genotyping. Okay, chips are available, SNP chips 9K, 90K, and now we are having 35K, 660K also. So these SNP chips can be used for genotyping, high throughput genotyping. It's very easy. One can get genotyped, and the advantage is there. Here, if you go for SSR, in case of wheat, we get say 10% or 20% polymorphic markers. So it's really difficult to get good coverage of polymorphic markers. But when you have SNPs, you get hundreds of polymorphic, thousands of polymorphic, large number of polymorphic markers you can get. So you can get more polymorphism with SNP. And now latest development is that wheat genome has been sequenced. So wheat genome sequence is now available, and that gives a lot of flexibility. We can develop primers, and we can design primers because we know the sequence. And at the same time, we know the physical position of any marker. We can simply blast into the genome database, and we can see what is the position. So these are the main genomic resources which are used. We can make use in our breeding program. And sorry. And this is some of the data I have given. It is only about 14 GB if you see here. Wheat genome size is about 17 GB. So this is chromosome wise total length in base pairs. So you can see 1A, 1B, 1D, then 2A, 2B, 2D. So these are the number of base pairs, but still it is almost 14 GB. It is not 17 GB. So there are certain things which are still missing. Some sequences we have to they have to complete it, but the information is now available, and this can be used. Now, first I will come off wheat rust, because this is the major disease in India, and we have been working for quite some time on wheat rust. And I will use wheat rust to explain the methodology of breeding, how we can make use of markers in breeding. So when we talk about rust, there are three kinds of rust: leaf rust, that's caused by Puccinia triticina, then there is stem rust, and then stripe rust. So all these three rusts are present in India. Stem rust is not a major disease nowadays. It is not of much significance. By and large, we have been able to control this disease. And the reason is because we are having good deployment of genes, stem rust resistant genes, and the disease is not a major issue. Leaf rust occurs throughout India. Okay. Stem rust is a disease of warmer areas, so you can find stem rust in central India and peninsular India. Okay, that's a major areas where disease may develop. Leaf rust you find from north to south anywhere, because this disease has this pathogen has a broader adaptability as far as temperature is concerned. Slightly lower temperature also disease may come, and it may come at higher temperature also. Stripe rust is a disease which you will find in northern areas only, in northwestern plains, what we call Punjab, Haryana, northern western UP and northern Rajasthan also, Ganganagar areas, all those, and then of course Himalayas. So these are the areas which are having lower temperature, and this is the disease stripe rust that develop when temperatures are low. And this is the time in the month of January, say in areas of Punjab and Haryana, temperatures are quite low, and if disease develop, then it can be devastating, because that is a stage when the plants are not grown up, and it may cause substantial losses at initial stage. Okay, and then it get a lot of time to develop, and the disease can multiply, inoculum can multiply, and it can spread to wider areas. So this is a very important disease in this part of the country. So we have to tackle all the three rusts. Stem rust as of today is not an issue, but leaf rust and stripe rust, these are important. If we see the dominant variety of this area, that is, if you know, SD2967, and that variety has become susceptible to stripe rust. We have to take care of stripe rust because that's important disease. And then I will just briefly tell you about type of rust resistance. When you are discussing wheat, then you should be familiar what kind of resistance we are having. Now, resistance genes, a number of genes have been identified, and those genes have been designated. Resistance genes can be in wheat are normally classified into categories like seedling resistance and adult plant resistance genes. Seedling resistance genes are those which show resistance at seedling stage. Okay, so we do screening at seedling stage under glasshouse conditions. So you can have seedling resistance genes. So those genes will be conferring resistance against rust at seedling stage as well as at adult plant stage. So nowadays those seedling resistance genes, they are also described as all stage resistance genes because they confer resistance at all the stages of plant growth. So these are ASR gene, all stage resistance gene or seedling resistance gene. Seedling resistance because they are confer resistance at seedling stage, but it does not mean they confer resistance only at seedling stage. Resistance continues till throughout the life of the plant. So seedling resistance or all stage resistance is one, and other is APR resistance, adult plant resistance. So there are certain genes which confer resistance only at adult plant stage. So what happens if you test those plants at seedling stage? They will be completely susceptible. Okay, they will not be showing any resistance seedling stage. They will be showing resistance only at when plants have grown up, adult plant stage. As plant grows, the level of degree of resistance increases, and adult plant stage they show very good resistance. And then the other terminology is major gene and minor gene. In wheat, you will come across if you see the literature. Major gene means they give very good resistance, high degree of resistance. See, LR24 is one gene. It gives complete resistance. There is no disease. Okay, very small pustules you will find, and then even if it's there, there will be necrosis, and the disease will not spread. Minor genes, disease confers resistance, but it is not enough to protect the crop. So it doesn't give that high degree of resistance. So moderate resistance you will get in case of small degree of resistance you will get in minor genes. So major gene, minor gene. Then there are genes like race specific genes. Certain genes which are race specific, they confer resistance against races. So a virulent race may arise, and it may knock down a resistance gene. Okay, and then there are genes which are we call race non-specific. Race non-specific means they confer resistance against broad spectrum of races. They are not specific race. They will confer resistance against all the races. Whereas race specific, they may confer resistance against certain races, may not confer resistance against other races. So race specific genes are there, race non-specific genes are there, and most of the APR genes, they are race non-specific, and most of the seedling genes, they are race specific genes. Okay, so they are race specific. So they give high degree of resistance. Seedling genes, you will find that they give very good resistance, but there is probability that over a period of time some virulent race may arise, and that may knock down the resistance gene. So these are the different kind of terminology you should be familiar. When you go for breeding for rust resistance, you should understand what kind of genes you are dealing with, whether these are race specific or race non-specific, these are major genes or minor genes, or these are seedling resistance genes or APR genes, and that will decide what kind of phenotyping you have to do when you are mapping genes. So this is something about genes, and then very often we talk about molecular markers, but sometimes we have very good morphological markers also. If you see here, there is necrosis at the tip of the leaf. This is known as leaf tip necrosis. Okay, this phenotype is known as leaf tip necrosis, and this is linked with a leaf rust resistance gene LR34. Okay, so varieties which carry LR34, they often show leaf tip necrosis. So this is a morphological marker that's linked with LR34, and this is another marker you can see here, what we can say pseudo black chaff. Okay, so you can see here the dark red color you will see on the chaff, so it's called pseudo black chaff, and this is used this is linked with SR2, stem rust resistance gene SR2. Now, as I was describing, LR34 and SR2. LR34 is for leaf rust resistance, SR2 is for stem rust, not stripe rust, it's for stem rust. Okay. Now, SR2 is again a race non-specific, still it's conferring resistance, it's race non-specific. Same is the case with LR34, it's race non-specific. And again, these are adult plant genes, APR genes. Okay. And these are the genes individually they may not be able to protect the crop. If only a single gene is present. So, they have to be deployed in combination with other genes. So, that you have to choose a different combination of genes you have to choose, and you have to devise your strategy what kind of genes we are going to have for durable resistance. Now, just a few examples I am giving you. Wild species have been used in wheat. A number of genes have come from wild species. Of course, there are other genes which have come from, which are native to wheat also. So, they have their origin in wheat itself, but there are other genes have been transferred from wild species. So, LR9, many of you might have studied, LR9 gene was transferred long back from Agelop's umbellata. This was the first example of transfer of a gene from an alien genome, alien species. It was transferred from U genome, Agelop's umbellata. Then LR19 is transferred from Agropyron elongatum, or it is also known as Thinopyrum ponticum. Name has been changed now. So, in literature you will find both the names, Thinopyrum ponticum or Agropyron elongatum. So, similarly, LR24 is another gene, LR28. Then this LR52 its origin has in wheat, and this is a gene which we identified in durum wheat, LRTRK, and we are using this gene in our breeding program. And LR45, this is a gene which is from sickle cereal, and we developed very good, very robust markers for this resistance gene in our lab. And so, this can also be used in marker stress breeding, but these are all, if you see, except this gene, these are from wheat, and the remaining are from wild or some related species. So, same is the case here. You see here adult plant resistance genes, LR34, LR46, LR68, and LR67 and LR68. These are the 4 genes which are what we call adult plant resistance genes, APR genes, and none of these genes is effective individually. It may not be able to protect the crop because the level of resistance is not that high that a single gene can protect the crop. But this gene is known as LR34 as far as leaf rust is concerned. Then it protects against stape rust also, YR18, then stem rust also, SR57. Same is the case here, LR46, LR68, and LR67 and LR68. So, these are the APR genes. They do not have major effect. So, individually they may not be effective. So, if you choose any of these genes, then you have to choose in combination with other genes, so that this can provide protection to the crop. Now, then these are some of the stem rust resistance gene. As I said, SR2, even today it is effective, but it has to be used in combination with some other genes. Individually it may not be effective to protect the crop. And then for stape rust also we are having a number of genes, YR5, YR9. YR9 is no more effective. Once upon a time this was very commonly present in wheat varieties. This gene is from sickle cereal, and if you remember popular variety of that time, PBW343 in India, you might have heard name of this variety, and this variety carried YR9. Of course, it carried other gene also, but it carried a segment from sickle cereal, okay, and that conferred resistance against leaf rust. That gene was LR26, then stem rust also SR31, and SR31 is the gene, if you are familiar with UG99, that particular race is virulent on SR31. But fortunately, this gene is SR31 is effective in India because we do not have UG99. That particular race is we do not have in India. And then of course you have other genes YR10 and YR15, and a number of other genes are also available. I am giving some examples where you can use these. Now, when we talk about marker state breeding, there are 3 steps. All of you know, I will be discussing only basic things today. First step is parental polymorphism. Why you require parental polymorphism? For background selection. So, you should have, if in wheat there are 21 chromosomes, okay. So, if you take at least 4 markers per chromosome polymorphic, then you require a minimum of about 84 markers. So, parental polymorphism is important because if you want to do background selection. And then foreground selection, all of you must be familiar by now. Foreground means the gene of interest, which the gene which you are transferring, you are having marker for the gene. It may be gene based marker or maybe linked marker, whatever it is, and with the help of that marker you are selecting the gene, okay. So, you are selecting the gene based on your marker, and then you can have background selection. Nowadays, now it is your choice, one can go for background selection, one may not go for background selection, but background selection helps us in recovery of recurrent parent genome, especially in when you are going for backcross breeding program. So, in backcross breeding program it is important because it saves time and accelerates the program. It normally may take 6 to 7 generations of backcrossing. You can achieve the same results in maybe 2 to 3 generations of backcrossing. So, background selection is important. Foreground selection, of course, you have to select for the gene of interest, and this I will now explain. I will take one example. This was our own work. So I will explain it. Our idea was to transfer gene LR19. Again, this gene is from wild species agropyron, and then eagle SR26. Again, it is from agropyron, and then YR10. So we wanted to transfer 3 genes for multiple rust resistance. So it will confer resistance against leaf rust. SR is used for stem rust, and YR is used for stripe rust. So we wanted to transfer at least one gene each for these 3 rusts. So we selected the genes LR19, SR26, and YR10. YR10 is not used in India. It's not deployed on. So we selected this gene, and it is very effective in India. SR26, there is no virulence globally against this gene. So this is very effective gene against stem rust. And LR19, there is only one virulence that has been reported from India. Otherwise, this is also an effective gene. Now, 3 parallel programs we are having here. The first, our approach was that this is the donor parent that carries LR19. Get F1, then you make another cross. You get F1 here, and here you get F1. Now, when you are having this F1 generation here, you have to make sure that these are actually your cross seed. Okay. Sometimes what happens, that selfing happens. When you are making cross, you get some self seed also. So it's always a good thing that you check the hybridity of F1. So this gene LR19 in F1 should be in heterozygous condition. Similarly, SR26 should be heterozygous condition, and YR10 should be in heterozygous condition. So you can check this, that all the markers are in heterozygous condition. Now, from F1, it's markers to backcross breeding, so you have to go for backcross. Now, in BC1, here BC1 F1, if you see here, you backcross with F1 you cross with SD2932, you get BC1 F1. Okay. Now, what should be the population size of BC1 F1? Bigger is better always, but there are limitations. How much population we can handle? And if we are having these 3 programs simultaneously, then we have to handle all the 3 at the same time populations. And then these are not the only varieties. We are having other varieties also in which we are working. So it depends how much material we can handle. Now, even if we say here, we make about say 300 or 400 seeds, and that's possible by crossing, in which in one spike you may get about 40, 45 or 50 seeds easily. So it's not difficult. If you make 10 cross 10 spikes, you will get about 300 to 400 seeds. So you get here BC1. Now in BC1, what you will be doing? The first step will be what? Foreground selection. Just basic things I am explaining. It should be clear to everyone. BC1, first you will be doing foreground selection. Now in BC1, what will be the segregation? Gene will segregate. Okay, all plants will not be carrying LR19. So theoretically, 50% plants will be carrying LR19 and 50% will not be carrying LR19. Those plants which are carrying LR19, those will be in, the gene will be in heterozygous condition. It will not be homozygous. It will be in heterozygous condition. Same is the condition here with SR26 and YR10. Then, if you are having say 150 or 200 plants which carries the gene, which plant you should select for back crossing so that you can get BC2. For BC2 you have to select. You are not going to cross all the plants. What should be your strategy so that which plant you select? So one way is that you go for background analysis of all the plants, and the plant which shows maximum recurrent parent genome that you can choose for back crossing. Okay, so that's one approach. Now, but again that is a huge amount of work if you are handling a number of genes. Second approach can be that okay, you go for foreground selection, but you simply select based on phenotype. Phenotype, you just have this recurrent parent and you compare it with, and plants which are closest to the recurrent parent, those plants can be used for back crossing. And the third approach which we use is that we combine both the things. To reduce the workload, what we do, just we select plants, 10 or 15 plants, which are very close to the recurrent parent in BC1. And then those selected plants which are very similar to a recurrent parent, then we select those plants and we do background analysis on only those selected plants, not all the plants. Okay. So what happens here, when we do this background analysis of selected plants, then we can select plants which is having highest background recovery. Among those which resemble to recurrent parent, we can identify a plant which is having highest recurrent parent genome, and that plant can be used. Now, whether one plant we should use or we should use more than one plant. Normally, we go for 2 to 3 plants, at least 3 plants, and the reason is that if you lose one plant for any reason, then everything is lost. So always you can select plants which are having highest recovery, recurrent parent genome recovery, and you can select maybe 3 plants. You backcross all the 3 plants, but you may advance only 1 generation. Remaining you can keep for safety, or if population size small, you can go for other. You can take second plant also. So here it is in BC1. Now BC1 recurrent parent genome, what do you expect? Average is 75%. It was discussed earlier also in one class. Average is 75%, but your objective is to get more than that. Then only it will accelerate breeding program. So when you do phenotypic selection combined with background selection, you should get a genome recovery which should be substantially higher than the average value. Now when you get BC1, then you go backcross and you go to BC2. Again the same step will be followed here in BC2. BC2, what you will do? You will raise the BC2 generation, and again there will be segregation. If you say LR19, this will be in heterozygous condition, SR26 will be heterozygous condition, and YR10, and then there will be plants which are not carrying these genes. So there will again we expect 1:1 segregation of plants which are carrying gene and plants which are not carrying genes. Plants which are carrying genes, the gene is present in heterozygous condition. So you get BC2 generation. Same process you repeat here. You do foreground selection. You just remove the plants which are not carrying genes, and those which are carrying, you select plants phenotypically which are similar to your recurrent parent, and then those selected plants you can go for background analysis, and you can select plants which are having highest recurrent parent genome recovery. Those plants you can select, and now if recovery is sufficient, you can stop here and simply go to BC2F2. Self it, and you can go to BC2F2. And sometimes if you are not very good in your maybe coverage of chromosomes or phenotypic selection, may not be, you may have to go for one more additional backcrosser. So that decision you have to take. So but in our case we decided that BC2 two backcrosses are enough for us. Now here we sell these plants to get BC2F2. Now BC2F2, what happens? There will be segregation of this gene LR19. It's true for all these 3 populations. Okay, here it will be SR26 and here it is YR10. Now in BC2F2 you will be identifying plants which are homozygous for LR19. Okay, the plants which are homozygous for LR19 you can identify. You can identify plants which are heterozygous for LR19 if your markers are codominant, and then plants which are not carrying LR19, those plants will also be there. So you can identify homozygous plants. Then those plants which are homozygous for LR19, again you can do background analysis, and you will find that there is increase in the recovery. Many of the loci which were earlier in heterozygous condition, they will come into homozygous state. So there will be increase in the recurrent parent genome if you do analysis. So now our objective was to combine the 3 genes. So one option is that you take these homozygous plants here, you take homozygous plants and make intercross. But what we did, because it requires 1 generation of selfing, we made crosses with BC2F1 here, and made cross between the 2. So you get LR19 plus SR26. Now here LR19 is in heterozygous state. You know, here LR19, because it is BC2F1 generation, here LR19 is in heterozygous condition. Here, same thing is here, SR26 in heterozygous condition, and if you go for YR10, it's also in heterozygous condition. So you are crossing plants which are heterozygous for the gene. So here this plant is also heterozygous when you are making cross, and this plant is also heterozygous when you are making cross. So there will be segregation here. So in segregating population here, this what we say nil F1, because these are now near estrogenic lines, because we have recovered background of the recurrent parent. So these we call nil F1. Now these here we identify plants which are carrying both LR19 and SR26. There will be segregation here because we have selected BC2F1 plant. Now if we make cross at this stage, then all plants should have LR19 as well as SR26. Okay, so just to save 1 generation, we made cross here, but otherwise you can also, here plant is homozygous, all the plants should carry LR19, here all the plants should carry SR26, you make cross and all F1s will be carrying the gene in heterozygous condition. So it's up to you, you have to decide what approach you want to follow. You can make cross here, but you have to, there will be segregation and you have to identify those genes. And then you get nil F1, you select plants which are having both the genes, then you get nil F2. Okay, same thing is done for making combination. LR19, SR26, LR19, YR10, SR26 and YR10. You can have all possible all these combinations. And then if you want to combine the 3 genes, then you can intercross here. You can this can be crossed with this one, and again you can combine all the 3 genes. Okay, so this is the approach, and normally we use in our wheat breeding program, and this approach we have used in a number of varieties. So SD2932 is one variety, and other variety is SD2733, in which we have transferred this gene. Here LR19 and LR24, the genes selected by us were different. Both are leaf rust resistant gene, but LR19 is linked with SR25, so it confers resistance against stem rust also. Similarly, LR24 is linked with SR24, so it confers resistance against stem rust also. So there will be simultaneously pyramiding of LR19 and LR24 as well as SR24 and SR25. So same scheme followed here, and we can pyramid those genes. So this is the basic methodology which we use in our marker state backcross breeding program. Now I just want to show the first step, as I said, discussed earlier, was that you have to do parental polymorphism. That's that you can do. You have to use large number of markers, and you identify markers which are polymorphic between donor and recipient parent. Now if you see here BC1F1 or BC2F2 here, this is the donor parent. Okay, so either plants will be heterozygous or they will not be having gene. Okay, all the plants will be heterozygous, whether it is BC1F1 or it is BC2F2. All the plants which are carrying gene will be heterozygous. But when you go for BC2F2, you find segregation. You get plants which are heterozygous, you get plants which are homozygous. These are the plants which are homozygous. And so you get plants which are, one minute. So you can select plants which are homozygous as well as heterozygous, but that you can get in F2. Now another important thing in marker state breeding, whenever you do, and that is the most important thing, is that what you have on your gel, on your computer, should be the same in the field also. Okay, so gel you are representing some other plant, and field some other plants is tagged, then everything will be changed. So that precision is very important. Okay, identity of every plant has to be maintained very strictly. That's very important for this marker state breeding. And this is again a combination of plants. If you see plant number 4, it carries LR19 and YR10. So you have to screen for LR19, then you have to screen for YR10, and then you can identify plants which are carrying both. Here you can see this is in heterozygous condition, and this is also in heterozygous condition. So now you can select plant number 4 because it is carrying both the genes, and then you can select this and you can get both the genes in homozygous state. And this is background selection. I think all of you know by now, you have idea about what's background selection. And just I want to give you these are the real data actually. Now how you calculate percent genome recovery? Number of homozygous markers plus half the number of heterozygous markers. So you just count the number of markers which are homozygous, and then how many are in heterozygous condition, and then you take half of that value. So that becomes your percentage genome recovery. Now if we see the advantage of marker state breeding, then you see here, if you see here, this is BC1F1, and BC1F1 itself we got 90% recovery. Okay, BC1F1 itself we got 90%. Average we say is 75%, but you should get substantially higher than average. Then there is acceleration of the program. So here we got, you can see we got here 90%. Then in BC2F1 it went up to 96%, and then BC2F2 again 98%. So this is how that in BC2F2 we were able to achieve at almost 98% in this particular case. And if you see other cases, then you will find that in case of SR26 it is 82%. Still it's not as high as this case, but here it is substantially higher than average of 75. So and then you can see it increased to 94, and then in BC2F2 it increased to 94%. Same is the case with third gene YR10. So in BC1F1, BC1F2, you find there is increase in the percentage of recurrent parent genome recovery. So you get substantial, and this is more than 90% or 95% is good enough to develop a variety which carries that gene. Okay, and here advantage you have seen it that in 3 generations BC2F2 you have transferred the gene. But then you need not stop your BC to F2. You are having large number of BC F2 plants. You go to BC to F3, BC to F4, and then cell multiplication, testing, and other things. Finally, phenotypic selection is also important. You have to you have to phenotype that you transfer. Sometimes what happens, if gene is not tightly linked, there can be recombination between marker and gene. So that situation may happen that in some plants you find that marker is showing presence of gene, but gene is actually not there. So that situation may also happen. And other thing which about wheat, that I am talking about wheat, which you may not find in many other crops. Most of these genes, many of these genes are of alien origin. LR19 agropyron, SR26, SR25 agropyron, all genes. So with these problem with these alien genes are that they are not present as a single gene. A segment comes, a segment of chromosome comes as such. So when segment comes, there are many other undesirable genes which may be present on that segment, and that may have some sort of yield penalty or other disadvantage. So when we select a gene, it's very important that the segment doesn't have any yield penalty. So LR24, as I said, this LR19, we transferred it, but it may have some, any alien gene may have some, we have to be careful. And other thing is that there is no recombination between alien segment and wheat chromosome. When it is heterozygous condition, there is no recombination. So when it's actually a translocation, alien translocation. So that much segment of chromosome, wheat chromosome is lost. That is removed and replaced by agropyron or whatever species you have used. Okay, so that particular segment of wheat is replaced by a segment from wild species. So that can have its own effect on the plant. So selection of gene is very important. LR24 gene, as I showed in other variety, is present in large number of varieties which are under cultivation. So obviously the gene doesn't have any such negative effect. LR26 was another gene which was widely used, and we did not, of course it had some disadvantages. Yield was high, everything was good, but quality was not good. There were some issues with quality because the gene came from sickle cereal and it imparted some stickiness to duff. That was an issue with this. So whenever you use alien genes, there may be, you have to be careful in selection of gene, that there is no yield penalty, there is no other disadvantage of using those. Otherwise, the approach is very, will be different. First, you have to reduce the size of the segment because it's not a gene, it's a segment. So it's always good if segment is smaller. Longer the segment, more undesirable genes may be there. So that's very important, that when you go for alien genes, you have to be very careful what genes you are selecting for your breeding program. Okay, so this is about APR genes. As I said, APR genes phenotyping is very difficult. So seedling genes we can do phenotyping, and even without marker, if we are able to do phenotyping, then we can make selection. But in APR genes, at seedling stage, they are, as I said, mostly they are susceptible, they don't show resistance, and they show resistance only at adult plant stage. So, and adult plant stage also, they do not give very good resistance. It's a moderate level, and it's under high disease pressure. You may find difficult to differentiate whether plant is resistant or susceptible. So that situation comes, and it becomes very difficult to select these genes phenotypically. So here markers are more important. If we are having good markers, linked markers, or gene-based markers, for LR34 we are having gene-based markers. So you can use those markers and you can transfer genes, because one gene is not very effective. So here we have used here, you see LR34, LR46 and LR68. Individually these genes are not very effective. Okay, they cannot protect the crop. But this is the line Parula, which carries all the three genes. So Parula shows very high degree of resistance. It's highly resistant to leaf rust, and this is because of these three genes. So we use this as a donor parent and then transferred these genes in SD2733. Then there's another APR gene LR67. This is the donor line, and same approach was used. What I explained to you: foreground selection, background selection, and same scheme. It's almost same scheme. So here it is more important that we can select. Now here we got lines, individual lines with LR34, lines which are having L46, lines which LR68, and line with LR67. Now we want to see what is the effect of single gene, what is the effect of two genes if you combine together, and if you put together three genes, what is the effect? What is the additivity? To what extent resistance increases? Because Parula is having three genes and it is almost near to immunity. Very hardly any disease is there. So these are race non-specific genes. Okay, so these are not race non-specific genes. These are individually they may not be effective, but if you combine 3 or 4 genes, then because they are race non-specific. So, there is very little chance that a breakdown will occur. So, these genes may be important for durable rust resistance. Okay. These are race non-specific, individually they may not be effective, but if you combine 3 or 4 genes, they are very effective, they can protect the crop. So, with this idea, all these 4 genes, these are 4 well characterized APR genes, and these were selected for transfer into these varieties. And of course, we have developed some of the markers for LR45. As I said, this is a gene from sickle cereal, and we developed markers, very robust markers, which we are using in our own program. And then we identified some gene for stem rust resistance, and we have mapped and markers. Of course, these are not very close markers, so we have to identify closely linked markers here. And then again, agile of spelterdis also we transfer several genes. Okay. So, we are having many genes actually from spelterdis, so we are taking up mapping of those genes, and you have to develop markers. Then another area is molecular characterization, where you can use molecular markers. So, this is the resistance genes. We are having almost 40 or 45 resistance lines which have been developed from Triticum militini. Triticum militini is a tetraploid wheat which is having genome AAGG. So, we developed those lines, integration lines, and these lines have been characterized using molecular markers. So, you are having original luxation of wild species, you can characterize, you can see integration points and what is the extension of integration. So, what you can see here is that there are multiple points of integration of Triticum militini. And the reason is that Triticum militini is having A genome which is homologous to A genome of wheat, it is having G genome which is again homologous to wheat genome and shows pairing. Good pairing. So, that is why you find multiple integrations here in all chromosomes. But one interesting thing which we observed was that even in D chromosome, because there is no homologous chromosome, D genome is not present in militini, but still some of the integration in some of the chromosomes 6D, you see 16, yeah. So, we got some integration even in D genome. So, it means some sort of pairing might have occurred between maybe G genome and D genome. So, molecular markers here can be used for characterization, and not only this, we are using molecular markers for characterization for tracing the integration segment in our program. So, molecular markers we are using in wild species. So, you can see, if you see there is one gene we have transferred from leaf rust for leaf rust, that is from agilosomography, and it produces same allele in agilosomography marker. Of course, these were not SSR markers, these were SNP markers. We did, I am not showing that thing here. So, we did SNP genotyping and then identified markers, and those SNP markers were converted into PCR based markers. Okay. And when we tested those markers in the original margraphy species and the integration lines, we got almost identical alleles in margraphy as well as integration lines. That showed that integration has taken from agilosomography and the resistance gene has come from agilosomography. So, but that I am not showing here. And another disease of importance is wheat blast. You must be familiar with, you must have at least, not familiar, you must have heard about this disease, it was in news. And again, the significance of markers, because we can select, there is one translocation, alien translocation, that is known as 2NS to AS. Okay, this is carries LR37, SR38 and YR17, these 3 resistance genes. This is a segment which has come from Agilops ventricosa. So, from ventricosa this segment came, and this segment also imparts resistance to wheat blast. So, simply you can screen your varieties or for presence of this particular gene or this segment alien translocation, and you can transfer also, because markers are there, you can transfer also in any variety you want to transfer this particular translocation. So, that is how marker state breeding molecular markers are important, and you can make use of these in not only selecting the lines which carries this particular, many of the lines might be carrying this marker because we are having diverse material. So, we can identify lines which are carrying this, and those should show resistance against wheat blast. And then genes for fertility restorer. This gene, the number of genes have been identified for fertility restorer in wheat and you see RF1 to RF7. These genes have been reported earlier. RF8 gene was discovered by us, and we have developed molecular markers for this. And in hybrid wheat breeding program, we are using these markers. So, this marker was developed by us, and it is very good marker, and we are using this for development of restorer lines, and it becomes easy, because if you transfer restorer gene, it is really difficult. Either it has to be in you have to have a male style cytoplasm in the background, only then you can identify. Otherwise, you have to, every plant has to be crossed with a male style line, and then progeny you have to test for whether it restores fertility or not. But if you are having a marker, simply you can screen with marker and identify plants which are carrying resistance. So this is the gene which we identified in one of our lines, and we developed markers, and now we are using in our hybrid breeding program. And again same approach is used, B lines and R lines, restore lines and R lines we have, which were susceptible. You can see here, this is yellow rust. You can identify how, you see the extent of disease here. This is a photograph from Delhi. So you see the extent of disease here, and these are the lines which are showing resistance, clean lines. So that's here you can use molecular markers to transfer resistance. So we are using the same approach which I explained to you for transfer of these genes in the restore lines and B lines and developing resistant versions. And then another area which I want to discuss is RST gene. RST gene are genes which reduce the plant height, and the green, these are also called green revolution gene. Green revolution came because of use of RST1 and RST2 genes. Dwarfing wheat came, and there is one variety C306 which is a very good variety as far as drought tolerance is concerned. That's very good variety for quality traits for chapati making. It's considered good variety. So ITC they just wanted to work on this, and we have used these markers. These are allele specific markers. So we used markers for this RST1 and these are RST2. So these markers used, and then we transferred. You can see here the dwarf lines here, and these are the lines which are not having the dwarfing gene. So this we have used effectively. Here markers we have used effectively. Otherwise, it becomes very difficult. Either you go after every F1 you go for F2, select dwarf plants, and then you go for, because heterozygous plants you find it's really difficult to differentiate the plants which are carrying genes and plants which are not carrying genes. So you can use markers, and with the help of markers we selected in large number of lines we have developed, which are, if you see morphologically, very similar to original variety C306 except dwarfingness. One trait I don't know whether you can observe it or not. If any one of you can see here, these plants, it's a little late, maybe 5 to 7 days late than this. You can see spikes have come out here. That you can see. Okay, so that's important when we have to see that in maturity also it should match the recurrent parent. And then finally I will summarize here, because it's a huge topic. I will not go into all those QTLs for heat and drought, and it must be it will be covered in many different topic lectures. But these are the major areas. You can see development of molecular markers. You have a genomic information, then you can develop molecular markers. Mapping, QTL mapping, association mapping, for all these you require genomic information. Then molecular characterization, I showed you, it can be used, and even for cytoplasmic characterization one can use chloroplast based PCRs, chloroplast based SSRs, and then marker state selection, marker state backcross-breeding, marker state recurrent selection. I did not discuss this because this is another area where you can combine a number of QTLs using marker state recurrent selection. Okay, so whether it's drought or heat tolerance, when you are having a number of QTLs you want to combine, then you can combine using marker state recurrent selection. And of course, there are other areas like quality and other things which I have not touched, but basic methodology remains same. You should have marker should be robust, and then you can go for background selection for recovery, or if you want to go for pedigree method, simply you can go for foreground selection, background is not required, phenotypic selection can be made. So molecular markers, they have or genomic information have enormous role in wheat improvement program now, and especially now we are having a good genome sequence of information is available. It becomes very easy for us to develop markers and design markers, and now we know physical location also of markers. That becomes very easy. Simply you know from on which particular chromosome at what base position the marker is present. So that becomes very easy. Physically you can determine physical location. So these are some of the areas which are of importance and related to genomics. And with this I thanks all of you and our funding agency ICAR, IARI, DBT, ITC, and then of course Indian Institute of Wheat and Barley Research. We get inoculum, rust inoculum and other things, different kind of support we get from there, and of course our entire team. We are having good number of students, SRFs and scientists, everyone is working. So it was just to give, idea was to give you a brief methodology, describe basic things rather than giving everything. Okay, so basic method remains same, whether you are transferring a gene or you are transferring a QTL, basic methodology remains same. And if you understand basic things, then I think you can you can devise your own program, you can devise your own scheme, and you can take this and how you want to proceed. So with this, I finish my lecture here. And anybody wants to ask any question? Present status of hybrid wheat. Yeah, you see, hybrid wheat basic requirement we have met, means you should have good CMS lines that we are having, and we are using Timofev cytoplasm. As I said, restores we have identified, we have developed markers for restores. So heterosis is the only problem. Now, for last 3-4 years we have been working, and now we are getting about 8 to 10% heterosis. So now that has to be tested. We are having 2 to 3 location testing now, but we have to go for multi-location testing to see whether we are getting stable heterosis. Even if we get 10% heterosis and with the seed rate of 70% or 60%, I think it should be commercially viable. So we are working on it, and hopefully we should have some good hybrid in future. Major problem is heterosis, yeah. And for that you have to make large number of crosses actually. That's the major limitation. Very few people are working. You have to have large number of crosses, identify heterotic crosses. So that's the major limitation. Other limitation is that how you make crosses. You require restored lines. People are having limited restored lines. Without restored lines, if you use any wheat, then F1 will be sterile. So for that you require large number of restored lines. And now we are in the process of developing number of diverse restored lines. And the gene pool is entirely different, which we are using for development of restored lines. So restored lines are entirely diverse from what other material, wheat material we are having. So we are developing restored lines, and once I think these are very advanced stage. Once we get those restored lines, hopefully we will test them and hope to get some good heterotic combination.
I have one common question here. As wheat is exaploid, contributed from 3 different species, A, B, D genome, then how to develop marker system in wheat, sir? Marker system, how to develop marker system in wheat?
You see, marker, you can have say most of the markers you find, they are basically linked markers, SSR markers, okay? And if you see the SSR markers in wheat, you will find most of them produce multiple alleles, 2 alleles or 3 alleles, and that is because there are 3 genomes, okay? But their primary binding sites are present on all the 3, okay, 3 genomes. So they can amplify the region, but the number of repeats will be different. So you get different sizes. Then you can see which allele is linked with gene of interest, okay? Which allele out of 3, which allele is linked with gene of interest. In fact, mapping part I could not cover here because it required time, but that we have done it. So you can see which allele is linked with out of 3. You have to see polymorphism first. You do BSA, and then you identify which are polymorphic markers. And in spite of that, it's possible because they produce different size alleles. Okay, A genome will produce an allele of different size, B will produce an allele of different size, and D genome will produce allele of say different. You may get 3 alleles, but in other variety you may get only 2 because the primary binding site may not be present in that particular genotype. So you can have presence absence, you can have codominance, all sorts of things are there. So it's not a major issue here. Okay.
No more questions? Thank you, sir. Thank you very much for such an excellent presentation. Students, I would like to remind you, he is one of a unique persons who utilizes both cytogenetics as well as molecular markers in crop improvement. You can discuss with him more maybe in the off hours. Now I request Gopal sir to present token of thanks to Dr. Vinod.
Not required for me. Thank you.
We'll break for tea. We'll get some